Assalamualaikum and hi, my name is Firdaus, S76211. So today I will talk about introduction part for spices experiment. Our group A, got cumin as our spices. Simple. Spices are important ingredients in food preparation because they enhance flavor, aroma and color of food product. Cumin powder is commonly used in cooking and contains unique aromatic compounds that contribute to its strong smell and taste. There are three objectives of this experiment. First, to observe the physical characteristic of cumin powder, including color, texture, aroma and appearance, in order to understand the unique flavor and sensory properties of the spice. Second, to identify the properties and quality of cumin powder as a spice by examining its physical condition, aroma intensity, and overall appearance, which may affect its suitability and acceptance in food products. The last one is to understand the importance and function of spices in food preparation and food science. This especially the role of cumin powder in enhancing aroma, flavor, food quality, and consumer acceptability.
Assalamualaikum and hi, I'm Nurhamiza binti Abu Bakar, S76691. I will explain about method and material. Material used in this experiment is ground spice sample, 19 ml saline, PCA, DRBC, TG agar, in dilution bottle in storage bag. The equipment used include analytic balance, stomacher, water bath, incubator, and anaerobic incubator system. We move to the method. First, 10 grams of ground spice were weighed aesthetically and transferred into a storage bag, and then pour 90 ml of saline water. The mixture was shaken thoroughly for about 5 minutes in stomacher to obtain the first dilution, which is 10 -1 dilution. Next, serious dilution were prepared by transferring 1 ml of sample into tube containing 9 ml peptone water until dilution from 10-2 to 10-6 were obtained. From selected dilution, which were 10-1, 10-2, 10-3, 10-4, 10-5, and 10-6 were transferred into straight-line pressure dishes in duplicate. Melted plate count agar PCA was poured into the plates using the pour plate method. The plate were mixed gently and allowed to solidify. Then the plates were incubated at 37 Celsius for 24 to 58 hours. For the anaerobic and aerobic, using DCPA agar, transfer 1 ml of dilution to the DCPA. Heating it at temperature of 18 Celsius for 30 minutes. After 30 minutes, cool it down before pour into plate. For aerobic, incubate at temperature of 37 Celsius for 48 hours, and for anaerobic, incubate at temperature at 37 Celsius for 24 hours.
First, I will compare the results among microbial group in variety of spices in this plate. Group 2, which use coriander seed, has only one mold growth, and the single mold is large, filamentous, and fuzzy. The single mold growth might occur due to its resistance towards the coriander antimicrobial effect, and the single mold might have the adaptive mechanism that efflects pump, as once the antimicrobial agent enters the cell, the pump will expel the agent out of the cell actively before it causes harm. Then the group 3, which use powdered cumin, has no mold growth at all, because this might occur because the concentration of antimicrobial agent like cumin aldehyde might be high and the bioactivity must be. Then the group 4, which uses chilean powder, has only one mold growth, but the single mold form is small and smooth and does not have filament, and it is not fuzzy. This shows that the single mold form is not much healthy as the mold form in coriander seed, because the mold in coriander seed was large and fuzzy. Then the group 5, which uses cinnamon, showed the highest mold growth, which is about 2 to 3 mold, and this suggests that high concentration of mold will present more in cinnamon compared to any other spices. Like, even though the cinnamon has powerful antimicrobial compound like cinnamon aldehyde and eugenol, some resistant molds can still grow effectively because of adaptive mechanism, and the cinnamon is being rich in nutrients. The cinnamon is rich in carbohydrate and nutrients like glucose, as the mold can utilize the glucose for metabolic activities, as the cell will take up the glucose and oxidize to form energy, and the energy produced will be used by the molds to grow and multiply effectively. Then the group 8, which uses cumin powder, has also had one mold growth, and the single mold form is large and fuzzy, and this mold might be resistant towards the antimicrobial compound of the cumin, and the mold might be able to grow large because the spice might not be effective enough to inhibit its growth. Now I will compare the result among the microbial growth in variety of spices in PCA plate. And the bacterial growth in the coriander seed in PCA plate is greater than the mold growth in the DRBC plate, because the bacteria can reproduce and multiply very fast compared to the mold, as the bacteria has shorter life phase and greater exponential phase compared to the mold. Then the group 3, which uses cumin 6 in PCA plate, the powdered cumin has more bacterial growth compared to the powdered coriander in the PCA plate. This is because the powdered cumin might be slightly richer in macronutrients like carbohydrate, protein, and lipid compared to the coriander, and bacteria are able to source more nutrients in cumin for better growth compared to the coriander, and the cumin might have lower concentration of antimicrobial compound compared to the coriander, allowing the bacteria to grow more effectively in the powdered cumin. Then the group 7 with black pepper powder sample in the PCA plate has numerous mixed population growth of bacteria, and this might occur because the antimicrobial effect from the black pepper, which is the pepperine compound, might be not enough powerful to suppress the bacterial growth and causes the bacteria to be resistant and able to survive. Then the bacterial growth in PCA plate is greater than the more growth in DRBC. Then the group 8 with cumin powder sample in the PCA plate showed numerous mixed population growth of bacteria. This occurs because the antimicrobial effect from the cumin is not enough to inhibit the bacterial growth, and the bacteria were able to survive, and the bacterial growth in PCA plate is greater than the more growth in DRBC plate. Now I will compare the results among microbial growth in variety of spices in TGE plate. The TGA agar stands for tryptone glucose extract agar, and this agar contains less nutrients compared to the PCA agar as it lacks certain growth supporting components such as yeast, which is rich in vitamin B. Then group 4 with chili powder sample showed very low growth of mixed bacterial population. This shows that the antimicrobial compound that present in the chili powder has inhibited most of the bacterial growth, and since the TGE agar has low nutrients, it further suggests that it might suppress most of the bacterial proliferation that are categorized as fastidious bacteria. In the group 7 with the black pepper powder, it shows the growth of the mixed bacterial population to be low, but however the bacterial population in the black pepper powder is greater than the bacterial population grown in chili powder. Now I will compare the results among microbial growth for aerobic and anaerobic microbes in variety of spices in DCPA plate. The DCPA plate is a selective plate for most species such as rhizophorus and aspergillus species. Then the group 2 result, which involves coriander powder sample, it has numerous growth of aerobic moles. The numerous colonies formed is yellow in color, and this shows that there is a lot of lactose fermenting aerobic moles in the coriander powder, because the lactose fermenter can break down the lactose into lactic acid, and the lactic acid will lower the pH of the medium, causing the colonies to turn yellow. Then for coriander powder in the anaerobic plate, it was found that several growth of lactose fermenting anaerobic mole. However, the number of colonies formed for anaerobic mole is less compared to the number of colonies of aerobic mole. Then the group 5 with the cinnamon powder sample, it had a several growth of aerobic mold, and the aerobic mold formed were white in color, which is the non-lactose fermenter. Then the cinnamon powder in anaerobic plate, it was found that there are very few growth of anaerobic molds, and the anaerobic mold formed were yellow in color, and they are lactose fermenters.
Assalamualaikum, my name is Nashika binti Nudi. My metric is S76813, and I will starting with our group data, group 8, which is for PCA, we calculate a weight average of 1.2 times 10 to the power of 7 CFU per gram. For the RBC, the count were less than 1.0 times 10 to the power of 5 CFU per gram, and for TGE, it was less than 3.0 times 10 to the power of 6 CFU per gram. Moving on to our spore formers result for group 8, both are aerobic and anaerobic mesophilic spore formers show 0 to minimal growth, giving a final value of less than 1.0 times 10 to the power of 3 CFU per gram for both categories. Moving to data 3, which present all 3 test results for the coriander samples. For PCA, group 1 coarse coriander powder shows 0 growth, while group 2 and 6 face extremely heavy contamination with TNTC result. Interestingly, for both the RBC and TGE tests, all 3 groups show excellent profiles, with count remaining well below the detection limits. Next, data 4 compares the cumin powder samples between group 3 and our group, which is 8. The data across PCA, RBC, and TGE shows a very consistent and clean trend for both groups, where almost all parameters successfully remain flat and well below the detection thresholds. In data 5, we look at the chili powder sample for group 4 and 5 for PCA. Group 4 hit an ideal count while group 5 stay low. For the RBC, both groups show a minimal fungal growth. However, in the TGS test, group 4 captured a significant flat sour spore count with the ideal range, where group 5 remained completely at 0. Data 6 display the result for black pepper powder from group 7 and 9, while the PCA, the RBC, and TGE profile generally maintain low baseline count. The contrast between the two groups highlight how different processing or blend of the same spice can affect the final microflora. Moving to data 7 for aerobic spore formers, all 3 groups show minimal to 0 growth. However, for anaerobic spore formers, group 2 hit a clear ideal range at 2.0 times 10 to the power of 6 CFU per gram, while group 1 and 6 remain very low. For data 8, looking at the group 3 and group 8, both groups show an identical very low contamination profile, where both aerobic and anaerobic spore counts remain flat and stay well below the detection limits. In data 9, for aerobic spore formers, group 4 started out with a clear ideal count of 1.1 times 10 to the power of 5 CFU per gram, while group 5 was low. For anaerobic spore formers, group 4 hit a heavy TNTC value higher than 3.0 times 10 to the power of 7 CFU per gram, while group 5 stayed at 0. Data 10 show the data for group 7 and group 9. For aerobic spore formers, both groups show 0 growth. However, for anaerobic spore formers, group 7 showed up significantly with a heavy TNTC value higher than 3.0 times 10 to the power of 5 CFU per gram, while group 9 remained completely clear.